everybody. Welcome. Thank you so much for attending our first Spark webinar. We're excited to show you how MBF Bioscience has integrated several software features that will make your Spark segmentation data fair. For those of you who do not know us, I'm Macy Heel.
And I'm Aiden Sullivan, and we're part of the Spark team here at MBF Bioscience, which is a subgroup of Spark Mapcore. Also on the line with us today is MBF Bioscience's Scientific Director, Susan Tappan, as well as data curation team representatives, Marianne Martone and Tom Gillespie. We're all available to answer questions in the Q&A portion of this webinar.
We'll leave the last 20 to 25 minutes for a Q&A session, but please feel free to ask questions throughout the webinar and we'll address them at the end. You can submit a question by simply clicking on the orange arrow button to open the GoToWebinar control panel. Find the question panel, type the question in the box, and click send. If desired, you can undock the question box for a better view of the questions and answers.
Now I'm going to go over today's webinar agenda. Macy and I are going to start by telling you what MBF Bioscience's role is in Spark Mapcore and how we expect your data segmentation will move through the Mapcore pipeline. Next, we'll discuss fair data standards, why they matter, and how you can make your Spark data fair at a file level. Following this, we'll give a brief overview of fair image segmentation and how, with the features we've developed in our software, it can help enrich Spark data sets. We'll then demonstrate Spark-specific annotation features and neuronal reconstruction strategies in Neuro Lucetta 360. Once we've shown you how to segment image data, we'll take the segmentation file and explore morphometric analysis available in Neuro Lucetta 360's analytic companion software, Neuro Lucetta Explorer. We'll end with some information about how to access a license for MBF Bioscience software so you can begin segmenting fairly today.
As Aiden mentioned, MBF Bioscience is part of Spark Mapcore. We're dedicated to producing fair data with our segmentation tools. We provide Spark researchers with segmentation software for neural, vascular, and anatomical image reconstruction. Our team provides on-site and remote training and support directly to the researchers that use our software. We're also available to discuss your multi-resolution imaging strategies and image segmentation protocols. In addition, we act as a liaison between research groups and Mapcore to help facilitate the movement of your segmentation data to the engineers that create the 3D organ scaffolds at the Auckland Bioengineering Institute. Our microscopy image viewer, BioLucida, will also soon host Spark image data on the Spark data portal.
So how does segmentation data flow through Mapcore, and at what point does it interact with other Spark cores? After creating a dataset on Blackfin, you will use MBF Bioscience software to segment your image data. To begin generating fair data, you'll access a Spark license for the software. You will define some information about the experimental subject itself. Then you'll segment the neuronal structures and anatomical regions that are relevant. By segmenting your image with MBF Bioscience tools, it is easy to incorporate file-level metadata and fiducial annotations. We will further demonstrate this in just a few minutes. Following this, you will upload your image and segmentation files to the appropriate dataset on Blackfin. Spark Mapcore's scaffold team can download your segmentation, fit fiducial information to the scaffold, and register the segmented data to the scaffolds. Lastly, the scaffold and segmentation can be displayed on the Spark data portal once your dataset is published, allowing anyone to explore your data. Additionally, the data is made available to SimCore to generate data simulations, which can further expand the utility of the experimental data. Let's explore one example of how a Spark group has utilized the multi-resolution image segmentation strategies our software can provide. This is a segmentation workflow that the John Tompkins Lab at UCLA has used to understand the structure-function relationships of cells within the stellate ganglion. Their lab has backfilled cells within the stellate ganglion and recorded electrophysiology readings of that cell. They then image cells at high resolution to easily distinguish the neuronal pathways and image the whole stellate ganglion at low resolution. They segmented the neuronal structures in a high-resolution image using Neuro Lucetta 360. Next, they segmented the stellate structure from the low-resolution image with fair vocabularies and Neuro Lucetta 360. We worked with the Tompkins lab to register the high-resolution neuronal reconstructions to 10x whole stellate image segmentations, putting the cells into perspective. Next, the lab was able to analyze the structure-function relationships using the morphological and physiological data. Lastly, the data will be registered to a stellate scaffold, allowing comparison within and between experiments.
Now that we've mentioned FAIR quite a few times, let's dive into what FAIR is and why it's important. FAIR is an acronym for findable, accessible, interoperable, and reusable. To be findable, the data needs rich metadata with explicit data identifiers. These identifiers must be searchable in a registry or index. Because we have built-in API access to the SciCrunch vocabulary databases within NBF software applications, we are able to write unique identifiers to the data file for each contour that was segmented. For example, here, the endocardium of the left atrium has an FMA ID of 7286. This ID and the entire URL to the parcellation where the term is cataloged is findable to both humans and computers. To be accessible, the dataset needs to have an open protocol with unique identifiers, and the metadata must be available after the data itself is not. Your segmentation process should be captured on protocols.io and should live with the dataset on Blackfin. To be interoperable, the data must represent knowledge with FAIR-approved vocabulary in a broadly understood language to allow other systems or software to make use of that data. Our link to SciCrunch allows for this. To be reusable, there must be sustainable metadata that complies with Spark data sharing standards. File-specific metadata is written to each segmentation file recording the information about the sample using NBF Bioscience software. I'll have Aiden show you what I mean. She's going to demonstrate how easy it is to capture this FAIR metadata for each segmentation file with just a few clicks in Neuro Lucetta 360.
Yes, let's switch over to Neuro Lucetta 360 now. So here you can see the program's 2D and 3D windows. To open your image, all you have to do is drag and drop it into one of these windows. The Spark metadata dialog will automatically open after you load your image in. There are two sections of the dialog. The first section on the left side here is for your subject information. From the drop-down list, select the species your image sample was extracted from and record the subject ID, sex, and age of the sample. Once this information is recorded, it will be stored in the segmentation data file. The saved metadata will automatically populate in the Spark dialog when it is reopened in NBF Bioscience software for additional segmentation. Also, if you are blind to the sex or age of your sample during your experiment, you can select this option from the drop-down list and then return to change the metadata after your segmentation and analysis. This sample behind me, the image behind this dialog, is a 10x confocal image of a section of mouse lung from the Thomas Taylor Clarke lab at the University of Southern Florida. So to populate my segmentation with file-level metadata, I would choose the species, mouse, input my subject ID, my sex, and my age of the sample. The second section on the right side of the Spark dialog is where you would choose the anatomy term list that you would like to annotate your image data with. Select the anatomical term list that is useful for your sample using the drop-down list for organ, species, and parcellation scheme or atlas. For this sample, I will select lung from the organ drop-down list. I will select mouse from the species drop-down. And the parcellation scheme, I will choose species independent because there's not currently a different atlas or parcellation scheme available for this organ. You may have noticed that there was a connection alert at the bottom of the window as I filled out this field. That is the software connecting to the SciCrunch database. Each time you fill out the dialog, the software will pull down the most recent up-to-date term list. Then you'll be able to start annotating. So let's click begin. The contour selection tab will automatically populate with the anatomy term list. You can scroll through and choose your term that you'd like to annotate with by using the alphabetically ordered list, or you can even search for your terms like segmental bronchus. You can click to choose the segmental bronchus, and then you have selected that term, and you can start contouring and tracing the features within your image. If you don't see the term within your list that you would like to annotate with, you can always click the Request a New Term button here. This will launch the SciCrunch InterLex dashboard. You can click Create a Term, log in, and then just fill out the term name and definition to request a new term. The SciCrunch team will help get your term integrated into the proper organ list. Additionally, you'll see these question marks within every feature of our software. They're the context-sensitive help links. If you click them, they'll launch the context-sensitive help for that tool, and you can get step-by-step instructions on how to use that particular tool. This is the case for any of the tools in MBF Bioscience software. Here's a brief overview of what I just told you about how to use the metadata tools within MBF Bioscience software. You can also see down at the bottom here what our data files look like when you do write this metadata to them. So you can see the subject information, including species, subject ID, sex, and age, along with the atlas that you use to annotate your image. Additionally, if you have used any terms to contour your image, the term name will be listed in the data file along with the unique identifier, as Macy was talking about earlier.
When we segment image data, we create the ability to distinguish between different elements that are captured in the microscopic images. By tracing data in a 3D micrometer point space, we can define relationships between neural structures and obtain meaningful measurements. We can then enrich neural reconstruction data by segmenting other features such as anatomical surfaces and by identifying discrete fiducial regions. If you define the anatomical features with fair, machine-readable identifiers, you'll enrich the data even more, allowing others, both humans and computers, to access and make use of this information. These additional segmented features will give context to the neural reconstruction and give insight on structure-function relationships, especially when the data is paired with supporting data from, say, RNA-seq or ePhys experiments. In the context of Spark as well, this will allow others to further explore the data on the Spark data portal in the form of organ scaffolds, flat maps, and simulations. There are many segmentation tools available to use in Neuro Lucetta 360. In the next portion of the webinar, we'd like to demonstrate a few of those to you. One of those segmentation tools can be used to automatically delineate objects within your image data. To begin, load your image into the program and access the relevant vocabulary list following the instructions that Aiden demonstrated earlier. For multi-channel image data, use the image adjustment window to isolate the channel that you'd like to detect objects in. Pick an anatomy term from the contour selection toolbar. In this example, we chose the segmental bronchus. In the Trace tab, open the Outline Objects tool to begin segmenting. Outline a structure by clicking a representative object in the image. Neuro Lucetta 360 will display a preview of the outline objects here in white. Use the Outline Objects modal window to filter detected objects by increasing or decreasing the sensitivity slider or excluding objects by size. When you are happy with the results, outline the object and repeat on different colored objects in your images. Repeat this process on more planes at an interval until you have fully represented the relevant structures throughout the image stack. The unique ID for each contour will be stored within the data file.
Another segmentation tool that can be used to enrich your FAIR segmentation data file is the cell detection feature. To start, click the Detect Cells option and choose whether you'd like to detect cells in 2D or 3D volume. Next, choose the first color channel you would like to detect in. Filter the cells by size, choosing the largest and smallest cell diameters. Now, preview your marked cells on a single plane of the image. You can see them as red circles on the screen. Then, you can filter based on pixel intensity of a chosen color channel. Select your marker and finalize to marker cells. Then, view in the 3D window. Here you can also make manual edits until your data accurately represents the image. So we've covered how to contour regions and mark cell bodies using MBF Bioscience's tools. Next, Macy is going to show you how to segment and edit neuronal structures in Neuro Lucetta 360.
Thanks, Aiden. All right, so here we have NL360 open again, and we have that lovely image from the Thomas Taylor Clarke lab. And here I really want to segment the fibers in the red channel. So it's a multi-channel image. I'm going to deselect my blue and green channels and sync this image up with the changes I just made. So now I can only see the red channel here. You can see this is a stack and it's in our 3D environment. To begin tracing trees, go to the tree icon to open the tree panel. You'll see here there are 3 tracing modes: Smart Manual, User Guided, and Automatic. User Guided is a semi-automated way of tracing, and that's what I'll start with showing you today. Under the User Guided Tracing Options box here, there are 3 methods to choose from: directional kernels, reverse crawl, and voxel scooping. The 3 methods are separate algorithms that approach detection differently, allowing the user to choose which method works best for their image data at hand. When you've selected your method, zoom into a region of the image you'd like to initiate tracing. As I hover over the image, you'll see that a yellow circle appears, and the diameter of the circle matches the diameter of the process. I'll zoom in a little bit more here so you can see that. Left click to initiate tracing, and you'll see a sphere is rendered, indicating that's the first point of that tree. And next, really easily, without moving or without clicking down on the image, I'm just hovering my mouse over in the direction of where the process goes. It will render more circles, again matching the diameter of the process. And when I'm happy, I will click down and it will fully render that tree in 3D space. I can trace in any direction, which is really nice. And I can also amend any data that you've created by hovering over here. Let's make a bifurcation point here. When I hover over an existing tree, a blue sphere will appear, indicating that that's where we'll make the bifurcation. I'll place a left click and I'll continue to trace this branch. You can also use automatic methods in the same session as both user guided and smart manual. So now that we know that directional kernels works pretty well in this image, we'll go over to our automatic tab. And again, you'll see the same 3 methods are available for automatic tracing as well. There are settings here. You can manipulate these settings to improve the tracing accuracy. Things like increasing the trace sensitivity and removing traces shorter than a certain micron. And then with a click of a button, it will begin to detect everything within that image based off the criteria that you established. In the lower left-hand corner, you'll see a status bar so that user knows at what point the software is as it's detecting those objects. And then instantly we have our trees placed down on the image data. Any element in the software that you detect manually or automatically can be edited at any time. And you can do that for trees, somas, markers, contours, varicosities, you name it. To start, to show you that, I will select this edit button here. And we can do things like remove whole trees for more of a coarse grain approach on editing and delete that. Or we can look at everything point by point and make individual edits to the points in 3D space. You can do things like connect, detach, remove, change the size of those points, etc. There are also cleanup tools that are available after you run an automatic trace that goes to areas where the program finds there may be an issue, maybe a small fragment, and allows the user to easily clean those up and make their way through their segmentation process. And here's just a little overview of what we just discussed for your reference later.
So once you've segmented your image, there are many neuronal and regional analysis available in Neuro Lucetta Explorer. Here's just a short list, and all the results can easily be exported to Excel. Let me show you how. We'll switch over into Neuro Lucetta Explorer, and we'll open up the FAIR data file that Macy and I worked on throughout this webinar, creating the segmental bronchus contours using the outline objects tool and the FAIR data terms, the FAIR vocabulary terms, as well as the dendrites that we traced using the automatic tracing method. You can see that in the Analysis tab of Neuro Lucetta Explorer, there are many different types of analyses available to you, and you can combine them based on what metrics you would like to obtain. In this demonstration, I'll show you how to obtain measurements about the processes enclosed in a 3D structure using closed surface analysis. We can use these metrics to answer the question, what is the density of fibers enclosed in this section of segmental bronchus? The first thing that we'll need to do is select the contours that make up the segmental bronchus and the dendrites that we trace throughout the whole image. Next, we'll select the anatomical panel and choose closed surface analysis. Then we'll select to analyze just the dendrites in the closed surface. As the program loads, it's going to determine which fibers run within the segmental bronchus contoured region and only get the length, volume, and surface area of those fibers that intersect with the region. It's not going to include anything outside of the region like this fiber or this fiber here. So now you see my data table, including the length, surface area, and volume of the fibers within the contoured region. If I select the volume column here and zoom in, you can see that the program is displaying just the fibers that are within the segmental bronchus contours. Again, you can easily export this data table to Excel by right-clicking and selecting Save to Excel. There you can do any further analysis on the collected data that you would like. To continue to get our density analysis, we will need to find the volume of the segmental bronchus region. So we'll select all of the contours that make up our region and choose the Markers and Region Analysis option. Select Contours and choose 3D Contour Summary and click OK. And that's going to give us the enclosed volume of the whole segmental bronchus. Then we can compare this to the volume or length or surface area of the fibers that run through the segmental bronchus that we were able to find using the closed surface analysis. Again, I can right-click and save this to Excel and further analyze the results and customize them to match the research goals that a group may have in mind. Here's a summary slide of the closed surface analysis and 3D contour summary that I just reviewed for your convenience.
MBF Bioscience is really dedicated to making Spark segmentation fair. We have plans to implement more features to our applications that will create more fair data. These include developing conversion tools to allow for more data types produced by Spark researchers to become fair, and writing fair identifiers to trees, vessels, markers, and sets. Our roadmap also includes implementing new segmentation tools based on the feedback that we've heard from the Spark researchers that we currently interact with. These include punctured detection algorithms in Neuro Lucetta 360 and co-localization analysis in Neuro Lucetta Explorer. Like you can see on the left here is the punctured detection that we're currently developing. We've also made improvements to our image montaging tool.
If you are ready to get started with one of our many applications, please email us to request a Spark license. You can contact us at sparc@mbfbioscience.com. We can also provide software training, support, image evaluations, and assistance with segmentation and analysis strategies. All right, that wraps up our webinar presentation, and now we can move on to the question and answer section of the webinar. We have about 30 minutes left for questions, and I want to remind you that Susan Tappan, as well as data curation team representatives Marianne Martone, and I'm not sure if Tom's on, we'll have to check again, but they will be here to answer your questions as well.
There are a few resources here on this Q&A page for contacting us, emailing us, Slacking us. We definitely want to get in touch with you and we'll answer some questions. All right, so Maurice, I may be hopefully saying that right, asks what species are already available for segmentation and is it possible to request new species to be added? Do you want to flip to the species that we have already that we query?
This is Sue Tappan. So we've built this list of species based on the known information that we have from current Spark researchers. So if you are a Spark researcher and you need an additional species added, reach out to us and we'll work with the curation team to get that added. We definitely want you to be able to manipulate and record the data and access the term lists that are necessary.
I have another question. Does the tracing work showed on the 3D window allow us to see the Z position of each point? And definitely. Let's open. In the 3D environment, you can just hold your left mouse button and move the data or the image data, and you can see in 3D space where those points are. You can also use things like the quad view, which will show different projections of the data. Here you have it in 3D, in your XZ projection, and YZ as well. And the Z position of each point is recorded in the data file as well.
And it's also available to be seen in the 2D window. If you select a single point and hover over it, it will give you the X, Y, Z, and diameter information for each point.
Another question here, for the surface area contour selection in the 3D window that gives information on the dimensions of the structures, do the contours need to be closed? To do closed surface analysis, yes, they will have to be closed contours, but you can still get some information about the open contours as well. So if we select contours, region, we can get details about the contours. And we can also do things like layer length analysis where you define multiple open contours, and the metrics that are provided for that is the length of the fibers that cross through those open delineations or those layers themselves. So there are a few analysis that you can do with open contours. Another question, can you create surfaces for many contours? We are in Neuro Lucetta 360. I don't have any contours open, but what you can do in the 3D window here is you can shell the contours, so you have a nice representation of the data for things like publications, and you can visualize the shell.
Dr. Kumar has a question relating to analysis in the bronchus, asking is there a way to measure the length of the arc by selecting curves both axially, i.e. the center line, and laterally. I believe for the bronchus itself, we would have to define, I think you'd like to define a center line for the bronchus itself, I'm assuming. And if that's the case, then we would amend the contours to map their centroids, and then we can map those in order to get that type of analysis. So we do have a mechanism to allow you to do that. You could do a vertex analysis or a polar histogram as well. If you are not currently accessing Neuro Lucetta 360 or any of the other software that we offer to Spark researchers and you would like to, please reach out to us. We want to get you using the software so that you can promptly and efficiently move your data through the Spark data pipeline for fitting to scaffolds. This data, if you have image data of any type that goes along with other experimental paradigms that you're after, we can help ensure that your data is captured and represented on the portal in as many ways as possible to make it a really rich experience for other people, including yourself and other colleagues and other interested researchers to explore the data sets once they're made public.
Hi, it's Marianne. It sounds like there was a question about what if the organized study is not in the drop-down list. If that's true, just please let us know. This is Pasang, if I'm pronouncing that correctly, and we will create a term list for you. If you could give us some lead time, it does take a while to assemble the terms and then make them available through Microbrightfield. Any
other questions? I think we're ready to wrap up unless anyone has anything else that they want to propose.
Can you show the Blackfin?
Absolutely.
Connection?
So here, next to vocabulary services, which is where you would access the dialogue that we talked a lot about today to add metadata and access your ontology list, there's also an icon here for the Blackfin portal. And when we select this icon, what we didn't show before this was you would sign in with your Blackfin credentials, and you can access all of the data sets that are available to you on Blackfin. And you can go through these data sets, and the important thing is you can download and upload files directly to a data set through this connection.
So we would recommend, if you're going to use this tool, that you go to Blackfin and ensure that the BIDS structure of folders is set up for the data set that you'd like. And then as you finalize data through MBF software, you can navigate to that file and upload it. It does not have to be an MBF file. It can be any type of data.
Andrew made a good point here. It should be the Blackfin platform, not Blackfin portal. So we will address that immediately.
That's true, because the portal is a very distinct location for the public access of all of the data. So we want to keep that explicit. So that's a good point.
Yes.
Antonina wants to know if it's possible to segment a nerve cross-section image and extract contours or labeled images. The answer is absolutely. There is an automated tool that was demonstrated, but we have a couple of other automated methods as well to outline cross-sections and areas of high contrast, which are normally present with these cross-section images that you're talking about. And so you could generate contours to label those images. And as you mentioned, they could be used for in silico modeling. So that would be a great way that data generated in the image segmentation side of things can be utilized for simulations and other types of modeling behaviors.
Thanks everybody so much for joining us today. And again, this presentation will be available to you on a few different sources, the biggest being docs.sparc.science. So look there and we'll send you an email with how to retrieve that as well.
All right. Thank you for joining.